Okay, welcome back, folks. Today we're going to transition from chemistry to biology to learn kind of like one of the founding principles of biology, the central dogma. And so today we'll be talking about DNA, RNA, and protein as a framework for understanding how drugs work. So this is what we're going to learn today, folks. DNA is turned into RNA through a process called transcription. RNA is turned into protein through a process called translation. And that is going to be our focus of what we learn today. Of course, DNA, when our cells divide and multiply, also needs to reproduce itself through a process called replication. But we won't be focusing on that during our time together. So let's walk ourselves through this and learn about all of these steps, as well as what is DNA, RNA, and protein. We'll start with DNA, folks. So DNA stands for deoxyribonucleic acid. We generally abbreviate it because that is a pretty long name. We think about DNA as the set of instructions for everything we are, how we look, how our bodies work. Every single function occurring in our body, our ability to digest food, is really controlled by genes in our DNA. And those genes are just individual sets of instructions. So if you think about your DNA as being a book of instructions, a gene is a chapter, for example. And those individual genes, maybe one is controlling your hair color, for example. And this DNA, since it tells our cells what to do, since it tells our bodies what to do, this DNA is found in almost every single cell in our body. The only exception is really red blood cells. And it's how we as humans pass on traits to our descendants. So for example, if in my DNA I have a gene for dark hair, it's likely that I'll pass this gene on to any of my offspring. Now, what is the structure of DNA? So DNA has this double helix shape that I am showing you on the right. We call this a double helix because it has 2 strands. So this is one strand, and that's a second strand. On the outside, we have a negatively charged backbone. So this kind of ribbon-looking thing here has a bunch of negative charges on it. And that's what's on the outside. Then on the inside of DNA is that code. If DNA is a set of instructions, inside this code, this is the words. This is how we understand what the instructions are. And the instructions are called bases. They are adenine, or A, thymine, T, cytosine, C, and guanine, G. And so by reading A, T, C, G, we can read out what the instructions of our DNA are. And DNA is a very specific set of instructions because each base only has one partner they match with. So A is always matched with T, and C is always matched with G. So let's take a look at the bases in the middle here, and I want us to find the green A bases. So here's an A, here's an A, and here's an A, for example. I found 1, 2, 3 different A's. If A is always matched with T, we should expect all of those green bases to be matched with red bases. And look, 1, 2, 3, yes, A is always matched with T. And because of this, we call DNA complementary, because A is always with T and C is always with G. So if we know the sequence of one of the strands of DNA, we can predict the sequence of the other strand of DNA. And that is DNA. It is our book, our set of instructions, and we can read those instructions through the order of the bases A, T, C, G. Next up, let's talk about RNA and transcription.